Welcome to Should Have Known It, where we present 15-minute videos of things you definitely should have known. Hi, welcome to Shoulda Known It. I'm Aoki, and today's topic is why axolotls don't scar. When you cut yourself, your body does something efficient and a little brutal. Within seconds, platelets clump. Within hours, a fibrin scaffold forms. Within days, fibroblasts, connective tissue cells, pour in and start laying down collagen as fast as they can. This rapid collagen deposition is what becomes a scar. It is a patch, not a repair. Your body chose speed over fidelity. This is not a failure of biology, it is a trade-off. Mammals, especially warm-blooded ones, are metabolically expensive. A wound left open is an infection risk and an energy drain. Evolution, across hundreds of millions of years, selected for fast closure. The scar is the price of that speed, which makes it all the more striking that axolotls, a vertebrate with a spine, a heart, a complex nervous system, found a different answer. The axolotl is Ambystoma mexicanum, a paedomorphic salamander. Paedomorphic means it retains larval features into adulthood, gills, aquatic lifestyle, a body plan that never fully matures in the way frog metamorphosis works. For a long time, researchers assumed regeneration was tied to this juvenility, a kind of frozen child biology. It is now clear that is not the full explanation. Regeneration in axolotls is an active, gene-directed process. It is a skill, not an accident. After amputation, the first thing that happens at an axolotl wound site is not inflammation, or rather it is, briefly, but it resolves almost immediately. Within 24 hours, something extraordinary assembles, a structure called the blastema. The board says blastema because this word is the hinge the whole field turns on. The blastema is a mass of dedifferentiated cells. Dedifferentiated means they have, in a controlled and reversible way, forgotten what they were. A muscle cell near the wound site partially reverts it, down-regulates the gene expression that made it a muscle cell, and becomes something more like a progenitor, a cell that could become several things. Cartilage cells do the same. Nerve-associated Schwann cells do the same. The blastema is a temporary return to developmental flexibility. For decades, the assumption was that blastema cells were fully pluripotent, like stem cells capable of becoming anything. The 2010 work by Eli Tanaka's lab at the Research Institute of Molecular Pathology in Vienna overturned this. Using genetic lineage tracing, tagging cell types with heritable fluorescent markers before amputation, they showed that blastema cells are mostly fate-restricted. A muscle-derived cell in the blastema will make muscle. A cartilage-derived cell will make cartilage. Dedifferentiation is real, but it is partial. The cells remember their lineage even while forgetting their position. That distinction matters enormously for medicine. It means you do not need a magic totipotent stem cell to regenerate a limb. You need the right cells to locally dedifferentiate, proliferate, and then redifferentiate in the right spatial order. The recipe is not create new cells from scratch. The recipe is teach the cells already there to temporarily forget, then remember correctly. Remove the nerves from an axolotl limb before you amputate it, and the blastema forms small and then stalls. The limb does not regenerate. This has been known since work by Marcus Singer in the 1940s. He showed that a critical density of nerve fibers is required to sustain blastema growth. But why nerves? What are they actually contributing? The answer, developed across the following 70 years, is growth factors. Nerves in the axolotl limb stump release a cocktail of signaling molecules, anterior gradient protein 2, NAG, FGF2, neuroregulin 1, that keep blastema cells proliferating. The nerve is not building the limb. It is acting like a project manager. It holds the site in an active, growth, permissive state long enough for the cellular construction to happen. Here is the elegant part. If you denervate an axolotl limb and then implant a bead soaked in NAG, just the protein, no nerve, the blastema resumes and the limb regenerates. You bypass the nerve entirely. The signal was the thing, not the anatomy sending it. This is the moment the field understood that regeneration might be pharmacologically accessible. You are not transplanting nerves. You are delivering a molecule. Human peripheral nerves release some of these same molecules. Neuroregulin 1 is present in human nerve tissue. FGF2 is present. The signaling vocabulary is not exotic. What appears to be missing is not the vocabulary, but the listening. Human wound tissue does not have the receptor expression, the downstream gene network to respond to these signals the way axolotl blastema cells do. The question is no longer what does the axolotl have that we don't? The question is what did we turn off? And can we turn it back on? In 2018, Sergej Novoshilo and colleagues, again in the Tanaka lab, published the first complete axolotl genome. It is enormous, 32 billion base pairs, 10 times the size of the human genome. Much of that bulk is repetitive sequence and transposable element expansions, the genetic equivalent of a library with a lot of duplicated pages. But the protein coding genes are largely familiar. A gene called PERX1 is expressed at high levels in axolotl blastoma cells and is required for their proliferation. Humans have PRUX1. It is expressed in human limb buds during embryonic development. It helps build the arm of a human fetus, and then its expression is sharply downregulated in adult tissue. It does not vanish. It silences. The axolotl keeps it on. We turn it off after we are born. The same pattern recurs across the regenerative gene network. Genes like ALDOH1 and 2, which controls retinoic acid signaling during limb patterning, genes like MICE1 and MICE2, which specify the proximal distal axis of a limb, genes in the WNT pathway, which govern cell fate decisions, all present in the human genome. All active during human fatal limb development. All heavily suppressed in adult human wound contexts. The axolotl is not running different software. It is running adult tissues on the developmental version of the same software. There is a shorthand researchers use for this. The developmental toolkit is still in the box. Evolution did not delete the tools. It put the box in a locked closet after the embryo was assembled. The axolotl just never locked the closet. This raises the obvious question. If regeneration is possible, if the genes are there, why did mammals evolve away from it? The most compelling current hypothesis involves the immune system. Mammalian adaptive immunity, which matured over the last 150 million years, is aggressive and scar-promoting. The same cytokines that rally T cells and macrophages to fight infection are the cytokines that drive fibrotic wound repair. Regeneration requires a quiet, tolerant immune environment, not a defensive one. There is evidence for this in axolotls. Their immune system, while functional, is comparatively tolerant. Axolotl tissue grafts transplanted between unrelated individuals are often not rejected at rates that would be unthinkable in mice or humans. Their wound environment is genuinely different, immunologically lower levels of pro-inflammatory cytokines in the early wound response, faster resolution of macrophage activity. The blastema gets a window of quiet, and it uses that window. There is also the metabolic argument. Regenerating a limb is energetically costly. An axolotl regrowing a foreleg expends roughly 40% more calories over the regeneration period than a resting axolotl. For a cold-blooded animal that is manageable, their baseline metabolic demands are low. For a warm-blooded mammal already spending enormous energy on thermoregulation, the added cost of full limb regeneration may have been a disadvantage under evolutionary pressure. Scar tissue is cheap. A new limb is not. And so we got the scar. Fast, cheap, effective enough to survive. The axolotl, sitting in the cold waters of Lake Xochimilco through millions of years of gentle evolutionary pressure, was never pushed toward our particular compromise, and now it is critically endangered. Fewer than 1,000 individuals remain in the wild. The animal that holds the instruction manual for one of the most transformative capabilities in vertebrate biology is disappearing from the one lake system it ever lived in. If you want to argue that mammals entirely lost the capacity for regeneration, the spiny mouse will make you reconsider. Acamis cahirinus, a small African rodent, can regenerate full-thickness skin wounds including hair follicles and dermis with no scarring. It can regenerate large ear punch wounds, closing holes through cartilage. When its skin is under tension and tears, the tear heals cleanly. No mouse in any other genus does this. Ashley Seifert at the University of Kentucky has spent the last decade characterizing the spiny mouse wound response. The key finding: Acamis wounds have a dramatically muted initial inflammatory response compared to standard laboratory mice. TNF-alpha and IL-6, the pro-inflammatory cytokines that normally dominate early wound healing, are lower and briefer. The wound site achieves a more tolerant immune environment. And in that quieter environment, fibroblasts lay down less collagen and more extracellular matrix that resembles the original tissue architecture. The spiny mouse cannot regenerate a limb. but it demonstrates that the scar reflex is not hardwired in all mammals. It is a tunable parameter. Change the inflammatory tone of the wound environment, and the cellular behavior downstream shifts. This is the same logic that is being applied in early human trials, not replacing human cells with axolotl cells, but attempting to modify the wound environment to give human cells permission to behave differently. The first generation of regenerative medicine, from the 1990s through the 2010s, was focused on scaffold and stem cell transplantation. Grow a trachea on a decellularized scaffold, seed it with the patient's own stem cells, implant it. This approach generated enormous hype, a series of clinical disasters, and then a more realistic recalibration. The scaffolds degraded irregularly. The stem cells didn't integrate reliably. The field learned that you cannot simply install cells and expect the body to run them correctly. The second generation, which is where we are now, is more subtle. Rather than transplanting cells, it asks, can we chemically reprogram the wound environment? Can we give the existing cells the molecular signals that a blastema would receive and have them respond accordingly? This is what groups at the Wyss Institute, at Mass General, and at the University of Edinburgh are testing. A 2022 study from Tufts University, Michael Levin's lab, demonstrated full leg regeneration in adult African clawed frogs, which normally cannot regenerate, using a 5-drug cocktail applied locally at the wound site for just 24 hours. A single day of treatment initiated a months-long regenerative process. The drugs targeted the inflammatory cascade, the WNT pathway, and progesterone receptor signaling. None of the drugs are exotic, several are approved for other uses in humans. The frog grew a leg that lacked a few small features but was functionally weight-bearing. 1 day of drug exposure, 18 months of subsequent autonomous regeneration. The wound didn't need to be told every step. It needed permission at the starting gate, and then the developmental program took over on its own. That result was published in Science Advances, reproduced independently, and it is now one of the most cited papers in the field. Limb regeneration is visible and dramatic, but axolotl spinal cord regeneration may be the finding with deeper implications. After complete spinal cord transection, a clean cut through the cord, axolotls recover full sensorimotor function within weeks. The severed axons regrow across the injury site, functional synapses reform. This does not happen in mammals, where spinal cord injury creates permanent scarring and functional loss. The key inhibitor in mammalian spinal cord injury is glial scarring. Astrocytes, the brain's support cells, respond to injury by forming a dense, proteoglycan-rich scar tissue that physically and chemically blocks axon regrowth. This scar actually serves a function. It walls off the injury to prevent spreading damage, but it also permanently blocks repair. The mammalian CNS bought containment at the cost of recovery. Axolotls do not form this glial scar. Their astrocyte equivalent responds to spinal injury with a bridging, permissive matrix rather than a blocking one. The difference traces partly to a signaling molecule called CSPG chondroitin sulfate proteoglycan, which axolotls produce in lower amounts and with different sulfation patterns that are less inhibitory to growing axons. Researchers at King's College London showed in 2023 that treating mammalian spinal injury sites with an enzyme that degrades CSPG chondroitinase ABC significantly improves axon regrowth and functional recovery in rats. The axolotl mechanism pointed directly to the therapeutic target. And then there's the brain. Axolotls can regenerate portions of their telencephalon, the forebrain, after surgical removal. Replaced neurons integrate correctly and animals recover behavioral function. In 2021, a team at the Research Institute of Molecular Pathology showed that this regeneration depends on a population of radial glia that remain mitotically active in adult axolotl brain tissue. Adult human brains retain a small population of similar cells. Whether those cells can be reactivated is the open question that is driving several current labs in a direction that 5 years ago would have seemed implausible. Honest projections based on the current publication record. Full limb regeneration in humans is not a 30-year target. It is probably not a 50-year target. The spatial complexity of a human hand, 27 bones, 34 muscles, 48 named nerves, is staggering, and the regeneration guidance system would need to be commensurately precise. But that is not what the next decade is actually chasing. The near-term targets are scar-free wound healing, functional nerve repair after injury, cardiac muscle regeneration after heart attack, and incremental spinal cord recovery. These are tractable, Heart attack kills cardiomyocytes, and they are not replaced. The dead tissue scars over, and the heart's pumping capacity is permanently reduced. If even a fraction of that muscle could be regenerated, the morbidity from heart disease, which kills nearly 18 million people annually, changes. That is the actual target the field is aligned around. The therapeutic strategy most likely to move first is localized, small molecule delivery to wound sites, the Tufts frog approach applied to human cardiac or nerve tissue. Not gene therapy, not stem cells, not anything requiring genetic modification of the patient. A drug applied locally at a defined window that tells the tissue to shift its repair program. This is achievable within the regulatory frameworks that already exist. Phase 1 trials for CSPG degrading spinal cord therapies are already underway in the UK and US. The axolotl does not know any of this. It is sitting in a tank in a laboratory or swimming in whatever remains of Lake Xochimilco, doing what it has done for 30 million years, losing its limb, growing it back, not because it is trying, because it never forgot how all we are really doing, all this enormously expensive, painstaking, brilliant science, is learning to read the instruction manual it has been carrying the whole time. There is a version of this story that ends badly, and it is worth saying plainly. The wild axolotl population is estimated below 1,000 individuals in the Xochimilco wetland system, a network of canals that was once a vast lake complex and is now partially paved over, drained for agriculture, and invaded by tilapia and perch introduced in the 1970s. Those introduced fish eat axolotl eggs and juveniles. The water quality is poor. The remaining habitat is fragmented. Laboratory populations are robust. Hundreds of thousands of axolotls are in captivity worldwide, but captive genetics are narrow. The Ambystoma Genetics Stock Center at the University of Kentucky maintains the primary research lineage, and it traces to a founding population of 34 animals collected in the 1800s. The genetic diversity is limited. For a research model, that is manageable. For understanding the full range of regenerative variation in the species, it is a constraint. Mexico has active conservation programs in Xochimilco, including refugia, walled canal sections where introduced fish are excluded, and water quality is managed. Some of these programs run by organizations, including the Universidad Nacional Autónoma de México, have shown genuine population recovery within their protected zones. But the surrounding habitat continues to degrade. The animal that may ultimately teach us to heal spinal cord injuries is listed as critically endangered on the IUCN Red List and has been for decades. The field likes to speak with confidence about mechanisms, and many mechanisms are now genuinely well understood, but the honestly unresolved questions are worth naming. We do not fully know what terminates blastoma growth, what tells the regenerating limb it is done, and prevents overgrowth. This positional information problem, how a cell knows where it is, relative to the whole body, remains one of the deepest unsolved problems in developmental biology. We do not know why regenerative capacity varies even among salamander species. The red-backed salamander can regenerate a tail. It cannot regenerate a limb. The axolotl can do both. The genomic and transcriptomic differences between these species are being mapped, but a clean mechanistic answer has not arrived. This variation is actually useful. It means regeneration is a spectrum, not a binary, and that incremental improvements in mammalian regeneration are biologically coherent, not a fantasy. And we do not know, not really, how the axolotl generates the correct spatial patterning in a regenerated limb. The blastoma knows which direction is proximal and which is distal. It knows to make a humerus, not a femur. It knows to stop at the shoulder. Somewhere in the positional memory of those dedifferentiated cells, the map of the body is preserved. Understanding how cells remember where they are, not just what they are, is the deepest question the axolotl poses. And it may be the most important question in all of developmental biology. Here is what I want you to leave with. The developmental toolkit is still in the box. PERX 1, Maze 1, ALD out, 1 at 2, the WNT cascade, the permissive immune response, these are not exotic axolotl inventions. They are conserved vertebrate biology, present in your genome, expressed in your body during the weeks you were being assembled, then silenced. The lock exists because it was useful, but locks can be opened. The last 30 years of regenerative medicine were mostly about finding the box. The next 30 are about learning to pick the lock correctly, not to recklessly reactivate everything, but to apply the right signal in the right place for the right duration and let your own cells run a program they already know. The axolotl is not a miracle. It is a reminder that this is, biologically speaking, a solved problem. We just forgot the solution. The animal is small, feathery gilled, slightly prehistoric looking. It was considered a god by the Aztecs, Xolotl, the god of lightning and death, who transformed into an axolotl to avoid sacrifice. The Aztecs may have misidentified which miracle it was performing. It was not cheating death, it was practicing resurrection. That's our time. The axolotl is patient. It has been doing this for 30 million years, and it will wait while we figure it out. Think about what it means to carry a capacity you've forgotten you have. The lights go down. We'll pick this up next time. And that brings us to the end. Thank you for joining me and learning something new. Until next time, click like and subscribe.